Hey everyone, so today let's talk about chapter 11, which is about not bankruptcy, as, as I've said as a joke a few times now, but about unicellular or one cell eukaryotes, okay? Which eukaryotic cells, as opposed to prokaryotic cells, are cells that have a nucleus and membrane-bound organelles, cells that we have in our body. Prokaryotic cells are things like bacteria. In this case, we're talking about protozoa, which are different than protists. Protists are unicellular organisms. When we talk about protozoa, we're talking about the animal-like organisms of the protists. So some protists look like this. Okay, you see as an example of a protist. In this case, this is a paramecium, and this is one of the many protozoa that exist in the world. So where did protozoa come from? So let's talk about the emergence of the eukaryotes, or the emergence of the protozoa, which led to more complex eukaryotic organisms like ourselves. Okay. The first evidence of life dates about 3.5 billion years ago, it says, and the first cells were very bacteria-like or more like um, prokaryotic cells. The origin of these complex eukaryotic cells came from what they call to be cellular symbiosis. Symbiosis is a close relationship between two organisms. There's different types of symbiosis that we'll discuss, but most likely this was mutualistic or mutually beneficial, or where both benefit, symbiosis among prokaryotic cells that led to the arise of these protozoa. So what they think happened was is they had a prokaryotic cell, and like it says on here, it pretty much was eaten into another prokaryotic cell and it became an organelle. So what they think happened is through this process, one of these prokaryotic organisms that were eaten by the other became the mitochondria, and the other one that was a photosynthetic bacteria evolved into the chloroplast. And this led to the rise of these complex eukaryotic cells that we have in our body that we've studied that have membrane-bound organelles as well as a nucleus. If we talk about protozoa in general, they lack a cell wall because these are your animal-like protists, so they're not plant-like or bacteria-like, so they don't have a cell wall. They have at least one motile stage in their life. This is similar to mobile, so it means that they can move. Okay? Most protozoa ingest and eat their food, but there are some that don't. Um, yeah, we already talked about endosymbiosis last slide, so we don't need to talk about that some more. When we talk about protozoa, since a lot of them are single-celled, they carry on all their life activities within that one single cell. They can only survive within a narrow range of their environment. So if they don't have the right amount of temperature, water, etc., they're not going to do very well, depending on which protozoa we're talking about. They're very important from an ecological standpoint. There are some very, very important protozoa out there, like Euglena, for example, which are photosynthetic. There's at least 10,000 species of them that are symbiotic. So they have relationships with plants and other animals. You guys might have heard of symbiotic bacteria before that live inside of your intestines that help you digest. So there's protozoa out there as well that are symbiotic and interact with plants and animals. And these relationships can be mutualistic, commensalistic, or parasitic. We'll watch a short video that explains this further, but mutualistic is where both organisms benefit. So mutually beneficial, you might have heard that term before. So both organisms benefit. The best example I can give you would be like the clownfish and the sea anemone. The sea anemone gives the clownfish a place to hide from predators and to live in, and the clownfish acts as bait for the sea anemone to get its next meal. There's commensalistic, where one organism benefits and the other is unaffected. This would be like the best example is sharks and these other fish called remora. Remora ride on sharks' backs and that allows them to not use up energy and they get a free ride. They also feed on the scraps that the shark leaves behind, and the shark is unaffected because it really doesn't care. Okay. Last one that you're probably most familiar with is parasitic. A good example would be like dogs and ticks. Okay, ticks just sap life energy from dogs, and dogs don't get any benefit from that. They actually suffer nutritionally. So that's a relationship where one organism benefits and the other suffers. This just shows you uh, some protozoa and where they are on this evolutionary timeline. So you have bacteria and archaea, which are ancient bacteria, and other bacteria, which are prokaryotic over here, so prokaryotic cells. And then here's your eukaryotic cells here, the most simplest being protozoa, which all of these are different protozoa, such as amoebas and euglenozoa. Okay, and then you have plants and you have different animals over here. So how do we define protozoan groups? It's based on some of their different characteristics and qualities. So you have heterotrophic protozoa. These are protozoa that obtain their organic molecules from other organisms. So they feed on other organisms, or they're basically feeders, right? They don't make their own food. They're heterotrophic. They get food from other sources. Some different types of these are phagotrophs, which these are heterotrophic protozoa that feed on visible food particles. When I think of this, I think of like Pac-Man, he eats the little visible food particles, right? So you have phagotrophs, and then you have osmotrophs, which feed on soluble food. So soluble would be like, it's able to dissolve in water. So I think of osmosis, soluble food, not actual visible food particles. Okay. Their mode of nutrition can vary. A lot of the times, like it says, it's opportunistic. So they are organisms of opportunities. They see an opportunity and they take it. They're not necessarily always hunters. They kind of are scavengers. Not scavengers in a sense like we say in ecology, but they find their food. Okay. These just show you some different protozoa on here. Not too important, just an image to add. Some other ways we define protozoan groups and classify them are their modes of locomotion, so how they move would define protozoan groups. Some move with cilia, some move with flagella, some, if they're amoebas, have their own unique types of movements. So let's talk about those locomotion movements. Like I said, cilia and flagella are two big ones. These are referred to scientifically as undulapodia movement. Cilia, the way you move is that cilia are bristle-like hairs that are all around your cell body. So what you do is you propel water parallel to the surface of the cell, since those bristles are all over your body. Whereas flagella, on the other hand, are whip-like long tails at the end of your body. So the way you move is you move water parallel to the axis of the flagellum, or where the flagellum is inserted into the cell body. So you can kind of think like a motor on the end of the boat has a similar movement. It propels water parallel to that motor. This shows you a flagella here. So it basically spins this around in a circle continually and propels forward. Whereas this with cilia, it's moving water parallel to its actual body. Okay, so to the cell surface in this case, it moves water all around, and this moves water just at the end, parallel to the actual axis of the flagellum. Some other forms of movement are pseudopodia movement. Um, this is used a lot in like amoebas. So you have lobopodia, which is large blunt extensions of the cell body. Like it says on there, those blunt extensions that are protruding out from the cell body contain both endo or inner cytoplasm as well as ecto or outer cytoplasm. You have Limax form, where the whole body moves as a whole, rather than sending out kind of an arm or an appendage like in Lobopodia. And then you have Philopodia, which are very fine, like the word F or the letter F here, or thin extensions that contain only ectoplasm at the end of the arms that are sent out. As I mentioned, the endoplasm is that inner cytoplasm that contains the nucleus and the organelles. The ectoplasm is that more transparent outer cytoplasm. Um, it's usually more rigid, and that's where the cilia and the flagella are attached to. This kind of shows you lobopodia right here. So you see this amoeba here. This is moving based on lobopodia. So see how this is a blunt extension. It's not fine pointed. Okay, so this is showing lobopodia movement, and you can see that it's extending not just the outer but the inner cytoplasm out to move as well. They talk about functional components of protozoan cells. This is nothing new. This is review from biology. Protozoa have a nucleus. Okay, you guys know what a nucleus is. It's the control center of the cell. It contains the DNA as well as the chromosomes, and it often has a nucleolus and chromatin. Some of these protozoa who feed through uh, phagocytosis, like it says up here. The way they do that is when they are, let's go back to this slide where it was talking about osmotrophs and phagotrophs for feeding, right? That's right here. Okay. So imagine like phagotrophs, phagocytosis. So you feed on visible food particles. And then the way that those food particles are absorbed in the body, once you fed on them, and this is review from biology as well, is when those food particles come into the body, they're put in a vesicle, or they're put in kind of like a small bubble that's surrounded with the cytoplasm. And then that vesicle that's formed, or that bubble, fuses with the lysosomes, which if you guys remember lysosomes from biology, those are kind of the janitor of the cell. They contain digestive enzymes, and they fuse with the vesicles, and they release those digestive enzymes, and that's how they're able to digest those food particles that are brought into the cell body. Okay. This is just showing you here how waste is getting rid of through exocytosis. This is something else you guys would have studied in biology. So here's a vesicle here. It contains waste. It moves up towards the end of the cell, towards the cell membrane, and it's flushed out. Endocytosis would look at the opposite of this image. Like we said in the last thing, where it forms those vesicles, the food would come in, it would form a vesicle, and then this vesicle would fuse with the lysosomes to be digested. We just talked about this, so excretion through exocytosis or pushing it out. Okay. It's also, it could also happen through diffusion where it just moves from high to low concentration and it's pushed out of the cell that way. Another cell organelle that protozoa have are called contractile vacuoles. These fill and empty in order to maintain an osmotic balance. Water enters through osmosis. So if you're a protozoa and you live in the water, you need to maintain a certain amount of water in your body, but at the same time, not so much that your cell's going to burst. So what these contractile vacuoles do is they leave a certain amount of water in your body, and they also continuously, as you see here, pump water out of the body as well, so that they don't get so large that they would burst, but at the same time, they don't dry up. I'm going to stop this here, and then we'll talk about the rest of the notes in the next lecture video. Okay, so this is part 1 of chapter 11. We'll pick up with reproduction in part 2 of chapter 11.